great honor to introduce the next speaker, who's my collaborator from Vanderbilt, Dr. Kevin S. Kevin is the, you can get his slides ready. He's the Gerald Pediatrics and Neurology, and he's also the Chief of Pediatric Neurology. He has himself a long-standing career interest in and our encounter the AHC years ago has kicked off a great collaboration that we're continuing today, even though we're not at the same collaboration. Is really a great way to make progress in science. So, Kevin.
Thank you, Al. All right, good morning. Thank you very much, organizers and all the families, for having us here. We are very grateful for the opportunity to present our work. I had the pleasure of following Dr. George, and I can't top that. So he gave some great background about AHC and where we're at, and I will share with you, I think, it's some very exciting results and new tools have been developed, which are really going to help drive this field forward. And this is about stem cell research we've done at Vanderbilt University. Now, go into a little bit of background what this technology is, what it means for both what we're doing now as well as the future. So let's see if we can get this answer to work. Like everyone, struggle. What's the trick? Really? Yep. Okay. So overview of my talk today. We'll talk again a little bit about what human stem cell research is, what it's not, the promises and limits, and we'll talk again about the work we've done at Vanderbilt as well as Northwestern University. And for the purposes of the talk, it seemed better to discuss how the cells are made and some of the applications that will come back at the end and discuss them. This is Vanderbilt Children's Hospital on the left, beautiful facility. That's Northwestern, Feinberg School of Medicine on collaboration, just like I think the AHC community is all about collaboration. Okay, so this is, and let me start with the one disclaimer or clarification. This is not embryonic stem cell research. This is a field that was developed only 10 years ago. This is an example how fast science moves. 10 years in science is a very short amount of time, but during that 10-year period where these first papers were published, the scientists who did this found out you could take cells from adults, even people of various ages, and you could take those cells to become what looks in every way like a stem cell. And I'll define what a stem cell means in a minute. So these induced pluripotent stem cells, or what I'll refer to as iPSC cells, have really taken over medicine and science, and it's really actually living up to the, and this work was so, after this was first described, the people who did this won the Nobel Prize, which is obviously a good consideration. But this is really a game changer. And the timing of it was also good, that when we were starting to get very facile with making stem cells from various patients, I was approached by both Al George at Vanderbilt as well as Kathy Sabota at the University of Utah at the time to be collaborators and put our interest and skill sets together. So this has worked really well, I think. So the promise and the hype, and this now is being delivered. Let me walk you through this slide as you can start to think very quickly how this will apply to AHC as well as basically any disease issue. Because again, we can now take cells from people, blue people apparently, and then they can be skin cells, they can be blood cells, different cell types which are differentiated to some specific function. We usually do skin cells because it's easy to get a small piece of skin and the skin grows back. It's quite straightforward. You do this reprogramming process, which I'll walk you through a little bit, 5 genes, and then take these cells and induce them to become stem cells, these so-called iPS cells. Now, these are the other downstream applications. Almost AHC research we'll talk to you about is really about this part here. Take a stem cell, which means theoretically it can divide forever and will be with us forever. That's a long experiment forever, but at least for years and decades, we have cells from patients we can freeze, we can thaw, we can FedEx them. So we have resources now, tools for the worldwide community to study AHC research applications. So what we want to do then is while you have stem cells, you can then make them, if you know how to do it. The application here is going to make them into human neurons, which are going to be different than a mouse, but have different strengths, different weaknesses. We can make neural cells, we can make blood cells, and we can use these, as we're doing now, to study the disease. What is disease mechanisms? Experiments that you can't human cells using human DNA, human gene. So it's all about the disease modeling. As mentioned before, drug screening has been used as well as other studies. And then what people often think about stem cell research, and while I think it's a great thought and it energizes me, we're not there yet. If you have cells from patients and you know what the patient's problem is, maybe you can use those same cells and give them back to the original patient to actually use it as a therapy. In AHC, in this hypothetical, really now still science fiction example, but it's a discussion to have, you could correct the mutation using genetic technology, which we're working on, to then put the patient's own cells back in if you knew how to do that safely, and potentially to give stem cells to other people as well. So you'd be laughed at 7 years ago, 10 years ago certainly was unheard of. Now it's a really rational scientific idea. So this is really, really exciting for all aspects of medicine in general. So as this is more of the spring 2016, there's many more cell lines in this. We have done skin biopsies or obtained fibroblast samples from Dr. Sabota from multiple patients, squiggly phenotype for their AHC aspects. We focus initially on the big three genetic mutations that comprise a large amount of patients. Also, we have the ATP1A3 gene too. And our goal of this is a repository of cell lines derived from patients with AHC genetic changes, and then be able to study those cells, study these neurons, and learn something about age, also learn something about AHC if you like the AO1N, and then even at the next level, and this is very exciting, this is where things are going, can you window it down to the individual patient? Because we're all different, we all have different genetic changes, different environmental, have a truly personalized medicine. And that's actually one of the strengths of my institution, Vanderbilt and Nashville, really building this initiative to have medical care being tailored to the individual as much as you can within reason. So this table here is just summarizing many different lines. They have code numbers, of course, for privacy issues, but they represent all the mutations, and they've gone through many levels of validation. If you start off with a skin cell, you need to make sure that this is a stem cell. It has stem cell properties, it expresses stem cell genes, it can then make other cell types, and I'll show you a few examples of that. So they're very, very rigorous validation is critical for these cell lines. And then some of the techniques we use, we also want to make sure that there's had any alteration. So a couple examples of this. So this is a picture, if you can see, well, these little spindly cells here, that's what human skin cells look like in a dish. I can take a skin biopsy from a patient, and we've done this dozens, hundreds of times now, and within 3 weeks, you can have these cells growing out of the dish. It's actually, this part's very easy to do. And then based on the other techniques, you can start making the stem cells in another 3 or 4 weeks. So in about 6 weeks, you can go from a skin procurement biopsy, excuse me, to having stem cells. Taking them to the next level and validating them takes probably another year or two. But again, once you have them and you freeze them, you're good theoretically. Unless your freezer goes down or you have a power outage, you're good. So one of the things we want to do again is be the repository for most of them already, but also ship them to other people, both as backups, but also so other people could participate and do their own research with these resources. The 4 names in red there are one of the examples of sets of genes that are normally expressed in stem cells. The reason these people won the Nobel Prize, they figured out the rules to reintroduce stem cell genes into non-stem cells, to again induce them to become stem cells. So that's just one example of the field of some of the genes that are used. They all have funny names, of course, but OCT4, KLF4, SOX2. And then this is an example, again, of what we're trying to do. on the left, this is the last time I'll mention it, but just to show you, if you have embryonic stem cells, they look like round, flat areas of cells. These are all human embryonic stem cells. These iPS cells that we've made look essentially exactly like the embryonic stem cells, and they seem to behave in our laboratory and many other labs to be almost exactly the same as embryonic stem cells. So one of the reasons this is a game changer, it's pretty much put all the concerns, whether it's ethical, political, religious, about embryonic stem cells is now almost a moot issue, because few people now are working on this. We're going right to the iPS cell system because it's easier to generate cells of interest. And if you have stem cells again, and I illustrate here with different organ systems relevant, you should be able to make any cell type you want, because that's what stem cells can do. It's a primitive cell, it can differentiate, and we have to have the rules and the tools and the chemicals to make cells become the right type of brain cells, let's say, or heart cells or kidney or what have you. And I think it is going to be important for AHC research, just as an aside, that we do not neglect cardiac cells too, given some of the phenotypes that are emerging. And actually cardiac cells, unlike brain cells, are actually very straightforward to make from iPS cells. So we could already go right from these stem cells with various ATP1A3 mutations and make any organ, and I think we should make brain and cardiac cells. There's now multiple methods of making these stem cells. This is a very quick history lesson. These various viruses, lentiviruses and retroviruses, and these are cumbersome to work with. This is what worked initially. This one was a Nobel Prize winning work, so it's so good. But pretty quickly, people want to move away from this. You don't want to take cells and introduce viruses to them, because that's the cells. Then you have to worry that this extra DNA may alter your results later on. So the next phase of this is all become non-DNA changing technologies. We've used that by introducing pieces of DNA temporarily called plasmids to these cells. So we inject these DNAs into the cell, they do their thing, they reprogram, and then they're supposed to be kicked out of the cell when they're done. And that's worked quite well in the vast majority of the cell lines we've made for using this plasmid technology. And then more recently, I'll show you just a couple slides near the end. We've also used a further advanced in technology, something called Sendai Virus. This is a virus that we all can get in the virus that does not stick in our cell technique, where you can introduce these viruses that, unlike these viruses on the left, which stay there forever, these viruses are very temporary, but you can very quickly introduce them to cells, they reprogram them, then the Sendai virus is completely gone. I'll show you data that we now have AHC lines made with this method as well. So anything can have different methods, different cell lines. It's all good to have these kind of tools available to us. Okay, so there's some examples. For any stem cells, they express various funny sounding gene names, nanog. There's a history to all these. There's an enzyme called alkaline phosphatase, or ALK-phos. It makes a pretty blue picture, and I'll show you an example later of AHC patient lines. But these are some of the markers that you know you're not dealing with skin cells anymore. You have stem cells. And they also have a very characteristic round appearance, and people who work in the laboratory have become very expert at picking these cells at the right time and not breathing at them or looking at them wrong, because they're very sensitive, human stem cells. But you get good at it, you can do this. And then this is some other characterization, just to get through some of the preliminary here. If you make a stem cell, any kind of cell you want on some level. So this is data showing that these stem cell expressing cells also can make, this is hard to see, muscle cells, this is endoderm derivatives, various part like gut cells, and these are brain cells, neurons. And the final assay you can do is you can inject these cells into a mouse, if it's a special mouse, and then make, again, all different types of human ectoderm, endoderm, mesoderm. These are the big tissues of the bodies that are one step beyond the stem cell. So these are all the assays, again, saying we've made stem cells, they're good stem cells, they do what they're supposed to do. All this work just to get to the start of the research. And then this is one plug for I think my second form of neurons that are the inhibitors of our brain, called GABAergic neurons. And this is my favorite pet hypothesis, that AHC has some significant issue with the inhibitory neurons, the one that tells our brain stop. And you can imagine if you have a problem with the stop or the go, who can have brain dysfunction. And that's just emphasized that one of the great things about having human stem cells to work with, it's not enough cells. You have to make certain type of brain cells. And I see the field already from talks this morning. We're now having good discussions about what are the chemical abnormalities in AHC. We talked about glutamate already. People mentioned dopamine, steroids. All those chemicals are made by different neuron types. We can now make all those neuron types individually and really test the idea which ones may be abnormal more than others. And this will really guide your therapy. You know, what are the really in the brain? Our brains are complex, right? So we have to think on the bigger picture. What are the actual cells responsible? What drugs can we use to target those cells only? Minimize side effects is how we treat. So this picture here in green is showing human neurons from patients' neurons. In the red, this is an older image. We're getting much better read. Are the specific GABA inhibitory neuron cells. Work here is shown again some of these experiments. Emily Craner did this work. She was in my lab, now child neurology resident at the University of Rochester. But she spent several months in the lab working on this project, and she was very excited and did a great job. But there is this example of the AHC that they express again, all these very beautiful stem cell gene patterns. One NANOG here, or TRA160. There's this alpha again I mentioned, this easy to do stain. And then she kind of went to town and just really made a lot of neurons and dishes and then make very beautiful pictures. But she made various neurons from different patients with AHC listed. So all these green extensions, these are the neurons that are actually reaching out and connecting to other neurons and forming networks in the dish even. So they formed that spontaneously. So we really then used her beautiful images to prove that all of these AHC lines were totally pluripotent and really backed up. And again, it's worth just emphasizing one more time, all these patient lines were selected because they had well-known ATP1A3 mutations to them, very well characterized, not only genetically, but also phenotypically. We know what patients these come from, we know what their quirks are, and we can start some of the description of their cells as well. Okay. So this is one experiment. I showed this a while ago too. One thing we're pretty good at is go to what we call neuroprogenitors. So these are cell types that are in our brain during embryonic development, where they grow somewhat, and then they're the cells that make neurons. So a progenitor cell is kind of the middle of the pathway cell. The stem cells, the neurons at the end, the neuroprogenitor. In 1 week, we can make, in this protocol we've optimized, about 80 or 90% of the cells can become neuroprogenitor. Neurons actually is much harder, and getting them in enough yield to do experiments is really challenging. But this is, again, I tell people this field 10 years old we started with making stem cells. So the fact that we're not quite sure yet how to make high yield of human neurons in a dish, we'll get there, but things do take a while to make these protocols work. But we have the starting material, and we know what we're trying to make, so we'll get there. And then for AHC example, there's a couple different, these stem cell markers are shown here in red. At baseline, just when they're sitting there, they're stem cells. They express high levels of this red stem cell marker. And then 1 week later, they show nothing. This blue here is just showing the cells themselves are there. They go from making this neuroprogenitor marker. So again, with AHC lines, we go from at baseline, there are no neuroprogenitors. 7 days later, almost have neuroprogenitors there, and even at green cells, which are actually neurons popping up too. So we can pretty quickly make the cells, but just back up one more time and think about what we're trying to do. We're trying to take a cell, it's a stem cell that becomes a neuroprogenitor, that becomes a neuron. How long does that take naturally in the human brain? That takes months and months and months to do that. So that process takes a long time. So one of the problems with this approach is we're trying to recapitulate Mother Nature, and Mother Nature takes months and months to make neurons. So these are a lot. To do a 100-day experiment is probably what it takes for some of this work. And there's one more picture of one of my favorite cell types, GABAergic neuron. And we use this assay to make fairly, not pure, but fairly robust numbers of GABAergic neurons in a dish. We want to start to do some of these functional experiments Dr. George will tell you much more about. But what we did in this next experiment is to take these well-characterized stem cell lines, make neuroprogenitors out of them, and then as much as we could make our neurons in a collection in a dish from either control patients who don't have any mutations to our AHC patients. Then we use a thallium flux assay again to start looking at expression of the protein and things that are altered. I'm good. So this is an example that within this dish, most of the cells there were neurons, and most of them were actually these GABAergic neurons, which was, this is a 100-day experiment again. And this is a very preliminary result, and this is using patient cells. John Snow, one of the students in the lab, no, that's J-O-N, this is J-O-H-N. He made these special neurons, then using the same thallium flux assay conditions, relative uptake of thallium, again, which is a potassium-like molecule used to look indirectly for the sodium-potassium ATPase. And this very preliminary experiment, but the difference here that these, again, are human neurons doing this experiment with, not some other cell type. The wild types nicely took up the thallium as expected, and then this other line, which has the G947R mutation, seemed quite impaired again. We have to repeat this. We have to try out different controls, different cell lines, but suggest that we can not only use cell types to screen drugs, we can also use human neurons to validate whether they work in these cells as well. So this is, to us, very exciting, and hopefully will continue to pay off. And then one other thing I'll show you as well, time working these neurons, and you need to make sure that they actually have mutations. My laboratory studies several diseases in addition to AHC. We have a lot of stem cells that we grow and make for other people as well in our laboratory, and we're human, so people make mistakes, and it's possible to switch lines around or people get distracted, things happen. Is to take the cells from the patient with a specific ATP, make the stem cells with months and tons of years. Then at the end, the stem cell you have that you're about ready to study and do all these other experiments to make neurons, it'd be nice to make sure that they still have the same ATP1A3 mutation. And this is easier said than done because the heavy-duty sequencing. So there's a couple examples that Grant Westlake, a technician in the lab, guy who loves this technique called PCR. That's all he does basically is PCR now. But he's made some protocols where we can take essentially this thing called PCR to amplify DNA, and then we can do sequencing. And this just shows you that these are, again, as mentioned in the first talk, these are heterozygous mutations. Of the 2 copies of ATP1A3, there's one change of the amino acid from one DNA change in one of the 2 strands of DNA you have. So we need to show that. That's what we have here. If you look, this is a typical chromatograph of DNA. Each color gives you a different letter of DNA, the A, G, C, T. And then what you see here in the middle mutation is actually a splice, which is not as common. See here they have a red peak and the blue peak, where every other one is individual. This is the mutation that causes this patient's AHC. Here's G947R. DNA has one clean color for one base, except for here, there's a doublet where there's the so-called wild type strand with the expected sequence, and then there's the one layer change, which is enough to cause everything. And now we validated all the lines we've had. To the next slide. Here you go. So just people are using these things. I just want to show quickly this Sendai Virus is a newer technology. It's pretty easy to use. You can essentially, from a company now, buy a little kit, and for several $100s, they'll send you this virus prepackaged with the reprogramming factors. And these cells that we use, the virus will go in, drop off its reprogramming genes, and then get out easily. The next slide. Okay. This time. So this is a very early Sendai virus. You can kind of get a hint. There's this little blue cell. That's the stem cells are not ready yet to be used. They still have this green expression of the Sendai virus there. But over time, again, it goes away. This says passage number 2. So we've grown them and passed them into cells probably a couple weeks after experiment. This is negative. It was passed 7 times after initial experiment. Now you can see that it's all blue, but there's no green, so the Sendai is gone. That's a good cell line. Then just to show it doesn't project in 1, there's no Sendai virus anymore in cells because they express this gene called AHC4 here very robustly. So again, I think this is helpful for the field. We'll be able to present cell lines that only from all patients, common ATP1A3 multiple ones, multiple patients, multiple versions of it, but also different techniques for making the stem cells. So in case there's some weird thing that happens, or how you made a stem cell from a patient's cells, you can get around that by using different methods. And all these things, theoretically, you can freeze and keep and pass, and so it'll be a tremendous resource. So where is this all going? I mentioned the early slide, I think, is the most direct application of stem cell technology is to do disease modeling using knowledge from individual patients to study their cells and to study diseases. And it really nicely complements. You cannot do research just in it. As awesome as mouse research is, mice brains are pretty different from ours. And if you do these two technologies together, you can really much faster figure out innovative strategies and therapies. Still want to validate them if possible on a mouse model, but having this stem cell line capabilities is really, we think, can accelerate all fields of medical research. The drug screening neurons, because they take so long to make human neurons, but you can still use them if you have secondary lines for screening or use computer programs to screen. And then it was mentioned earlier as approach. And then I forget if we mentioned in the back, we're starting a technology, CRISPR-Cas9, where we can not only take the cells from patients, we can now correct them. We can do an enzyme therapy, which is becoming, nothing's easy, but it's doable to actually then correct the DNA on the cellular level to a wild type. We're taking cell lines we've made from volunteers, so-called normal people. I don't think anyone's normal, but normal people. And then with their cells, we're introducing an ATP1A3 mutation into their DNA. And then that way we can, and with those really well-characterized cells from control volunteers, make sure that when we make this DNA change of any variant you want to talk about, we can then see whether the pathology, the changes are then introduced in normal cell types. So it's really using genetic engineering to both do therapies as well as and genes. And that gets very quickly, I think, don't get excited of, gosh, can we use these cells as a therapy someday? Can we give them back to people's brains? And I'd like to see my career at some point, yes, but it's hard to know. Again, 10-year field, let's see what the next few years gives us. So I want to thank the work. I mentioned Grant and John and Emily as well. Excellent collaborators with Dr. George, Simmons, and Sabota. Thank you all for including me. It has been tremendously generous. And I thank all of you all the time for the work you do to help support us. And I think this is a great journey, and hopefully we can get things figured out as quickly as possible. So I'm going to pause there. I can take any burning questions. Dr. George has a few more slides to talk about how he's used some of these cells to study things, or we can just kind of do a little discussion at the end, whatever people want to do. Okay.
I hate to be the only one, but the one thing with the stem cell, I mean, I could see where you're going to try to find some way to prevent the paralysis and dystonia and all of that, right?
Mm-hmm, correct.
And find a medicine or whatever. But the effects that are already there, you know, when like my kid, 24 years old, nothing's going to undo that, right? The learning deficits, the crookedness in walking.
Yeah, so I think your question is a little bit outside of the stem cell research, but we know from other diseases that even adult treatments can change things, and that's been a big lesson. There's a disease called Rett syndrome, which everyone has the same thought as you did, that once you're a certain age, that's that there's severe changes. But in the mouse now in clinical trials in kids with Rett syndrome and young adults, our brains are more, you know, responsive than we think. You can actually change quite a bit even when you're older. So I would keep some optimism that the brain can recover to some extent, but I agree with you in the big picture. The more there is, the longer it goes, probably the less reserve you have. But I don't think it's zero. I think people can still have some improvement, and I think we need to keep open minds that maybe we'll find something that's fantastic and unexpected that, oh my gosh, if you just throw this switch, these neurons can be made. And the other thing that I didn't want to get into, because it's a little different topic, but it is related, you know, we make neurons in our brain, new neurons all the time. And that helps us with memory and learning, and that was something we never thought was possible. It's very clear now that humans make new neurons as adults. So if we could even affect that population, maybe that would still help people who are more advanced by, you know, helping their natural abilities be augmented.
Oh,
so I would see. The question in the back. Oh, microphone on. You know the rules.
I just wanted to add what he was talking about is neuroplasticity, and I got a book to recommend on neuroplasticity. It's called Soft Wired. So if you want to read about that and how plastic the brain is, and when you fix the problem, there is hope for change.
Yeah, but in general, plasticity, yeah, younger the better. So
yeah,
we can identify
absolutely.
Yeah. Again, we don't know what the upper limit is yet. Yeah. So Dr. George is still the chair of pharmacology at Northwestern.
I think we don't. Yeah, so I thought we decided to tag team this only because it made sense to show you sort of the temporal flow of the cell generation in Kevin's lab, and I got to tell you, he does a very high quality job of checking everything very compulsively to make sure they are right. The cells then are now shared with us in Chicago, and we've been analyzing neurons that his lab has made and sent to us. We've also locally generated some neurons and have recorded from them, and our goal is actually now to study them for their electrophysiological properties. Do they fire action potentials, and are there properties of their are there neurophysiological properties that are distinct in the cells made from the patients with AHC from patients who don't have AHC, what we call normal controls. So anyway, there is another slide after this somewhere. Bill, are you queuing up? He's working on it. To escape out of it, and then we can just reboot it. Anyway, the technique that I'll be illustrating for you is, we call it patch clamping. Doesn't mean anything to you, but this is a method where we take a single cell, we take a small capillary tube, draw it out to make a tiny end, something like 10 microns in diameter, and then that is placed on the membrane of the cell, connection with the cell, and then using some amplification, we can actually record individual electrical events in the cells. And so what I'll be showing you is actually action potentials, the things I started talking to you before. So that's a Chicago skyline. Looks like that every day. This is some pictures of some neurons. There really are three classes of neurons that we have been focused on: excitatory cortical neurons, GABAergic interneurons, although we don't have data for you today, Those were Kevin's favorite cells, as he mentioned. And then lastly, I'll show you just some things we've done with spinal motor neurons. The reason we did that is that it was an easy protocol that was very robust. And so the first thing I'll just show you is this the timeline of making the neurons. It takes about 3 weeks to make them. They get plated on, or they get seeded onto a dish that has mouse astrocytes, a supporting cell from the brain. And here's some pictures of those neurons. The ones in red are the actual neurons. And here are some action potentials that we recorded. On the left are neurons, are action potentials that were recorded from a person without AHC, is someone with AHC who has the G947R mutation. These are both female patients or female subjects. And what's not clear, of course, what you're seeing here is there is a time along the bottom, left to right, and then the height of these peaks is really the voltage in the membrane. And so these spikes that go up fast is an action potential. And there is a stimulus applied, it fires an action potential, tries to fire a second one, and then there's just really not much oomph left in the cell. And that's pretty typical of this kind of neuron. What is different is probably what you're wondering. Well, look here. This is a very subtle difference. Here we have the baseline membrane potential, or what I will call the resting membrane potential. That's that fundamental charge that's put on the neuron in part by the action of the sodium-potassium ATPase. It's almost minus 60 millivolts in the control, 5 millivolts in the AHC patient. So there's an abnormal resting membrane potential, which we would have predicted. There's going to be a less contribution to the resting membrane potential because of that. The other thing that we see is the height is blunted. It's not getting up as high as the other. And that's also something we would have predicted if the sodium gradient isn't as strong and that the amount of sodium that rushes into the cell is less. So these are two things that we noticed. This is a compilation of lots of cells. Those were single cells that we recorded from, but Chris Thompson in the lab, who made these recordings, generated data on nearly 20 cells from each group. And here it's just simply if we add in more and more current left to right, you get more and more action potentials. And there's a subtle difference in here. We have to do some more analysis. But on the right is the real major observation, which is this resting membrane potential is less negative in the cells. Again, predicted a little bit based on the loss of the ATP1A3 activity. Now, this was just one pairing of one patient and a control subject. We have another cell line that we got from Kevin's lab from a male patient with the same mutation. And the question is, do we see the same thing? It's a different experiment, different person, but the same mutation. And in fact, we do. Here is the female patient that I just showed you. Here's the male patient. The trend is the same. There's significantly different levels of resting membrane potential. The absolute values are a little different, and that we'll have to sort out whether that's a true difference or not. So there's clearly an abnormality in the resting membrane potential. Another property that's different, and this is a more complicated property, is the amount of electrical current we have to inject into the neuron to get it to fire an action potential. That is called the rheobase current, and it's essentially just us dialing in the volume control on the experiment. And the amount of current that we have to inject is greater in the mutant cells by a little bit. It's still, it didn't in this experiment shake out to be significant, but in another experiment it did. So those are two things that we found that are fundamentally different. Next step is going to be to look at some additional pairings of patient and control, look at some other mutations, and then begin to move on and look at the other types of neurons. For example, we decided we would look at motor neurons. Motor neurons was just an easy way for us to get started. We didn't really expect anything, but we found a difference. Motor neuron potential. So you can see here when we inject a little bit of current, you get a flurry of action potentials in the control subject. But in the mutant line, we don't see anything. There is a resistance to firing. Now, admittedly, we need to repeat this a few more times, but we found this to be a very dramatic difference, and it began to give us thought about, well, why is this? Well, if we look at the resting membrane potential now, which is on the right, instead of being less negative, it's actually more negative. So the motor neurons behave very differently. So I think the only reason I'm showing this is to, number one, illustrate progress. Number two, illustrate there's heterogeneity among neurons. No big surprise. And third, to get the clinicians in the audience thinking about, is there motor neuron dysfunction in this disease that we've sort of overlooked or that's been masked at a central nervous system defect. So this is a single motor neuron in culture. It's not in a patient. So anyway, again, a collaboration with Dr. S's lab, we've been able to use neurons from these stem cells derived from AHC patients, and so far clearly have a less negative or depolarized resting membrane potential, very much in line with a loss of function of ATP1A3 in those cells. And we think it'll pan out that those neurons will actually require a stronger stimulus to activate. A curious observation on motor neurons that I think we will follow up when it's time. So anyway, that was the second part of the talk. So. Now we are about on time. I think the next talk, who started at 1:15, we're right about there. And I would recommend we just move forward unless there's maybe a couple burning questions.
I have a burning question. Siggy from Iceland, wondering if, and I don't know if this is something that you have worked on, that there is a GABA deficiency with the that have been with the mutated mice, and GABA deficiency has a lot of symptoms that are very much like. And GABA has to do with the sodium-potassium pump. It doesn't work unless it has GABA. So have we tried to get the to see if the mice will work? I know it's difficult to get GABA through the blood-brain barrier, but still,
well, I think maybe we can talk offline a little bit about what you're citing, but GABA is a neurotransmitter. It's made in neurons. It's released as a chemical transmitter to stimulate the adjacent neuron. The way the neuron amino acid and converts it, it doesn't reach the brain. It's not how it gets there. So giving it to mice would not really have any effect. So again, I think there is almost certainly a role for GABAergic neurons in this disease, which I think there's some evidence from McCarty's mouse model that, but I think there's a lot more to learn. But again, we haven't done a GABA therapeutic experiment yet.
This question is for Aaron a little bit later.
Okay, all right.